It is my great pleasure and honor to welcome our first speaker, our first keynote speaker, and that is Francesca Cotrufo. She is a soil ecologist who focuses her work on litter decomposition and the dynamics of soil organic matter. She's currently a professor and associate head in the Department of Soil and Crop Sciences, as well as a senior scientist at the Natural Resource Ecology Lab at Colorado State University. And her talk is entitled Advancing Understanding, Quantification and Forecasting of Soil Organic Matter Dynamics to Inform Climate Change Mitigation Strategies. So, Francesca, please.
Challenge of being short. Thank you all for being here, and thank you for having invited me to this great meeting. I'm a soil ecologist and biogeochemist. I decided today to give you a very broad overview of what we do in my lab. And unfortunately, I will have to leave you guys tomorrow morning as I have a busy teaching schedule in the fall. But please grab me a lunch or any time to go in depth in any of the field I will touch. So first of all, why we are all here, right? It's a pretty challenging time for humanities, and if you guys have kids like I do, it's, you know, we are thinking seriously about the type of planet that we are going to leave to the next generation. You know, global population is reducing its growth rate, but is still growing and having huge demands on our planet. And the fact that we actually do a very poor job at using resource with an equitable approach, that put even more pressure on a planet from which we need to harvest food, we need water, we need energy, and that put a lot of pressure on our lands. And on top of that, we have humongously increased the emission of greenhouse gases to the atmosphere so that the planet is hotter, and that is getting agricultural even more challenging and producing those resources even more challenging. So there is no more time to waste, as many of you have worked on this for 30 years, and we keep saying the same thing. And now we are really at a point in which we cannot waste any more time, and we do need to not only halt the emission of fossil fuel as soon as possible, but we also need to draw down carbon already in the atmosphere. And that's where actually soil come about, but not just on climate. Soils are really at the nexus of all these challenges that humanity is facing. That's the place where the large majority of our food come from. Soils recycle water and depurate water and so forth, and of course are central to sequestering carbon and contributing to climate change mitigation. And there have been a number of studies, this is just one, that has tried to estimate the potentials that soils have with respect to sequestering carbon out of the atmosphere. And some of those approaches have different numbers than others, depending on whether they occupy more, they can be done across more land or have more potential per unit land. But overall, there is a significant capacity from our soil to sequester carbon if we modify our management. And the other thing that is important to point out is that today there are several different approaches that can be followed to sequester carbon out of the atmosphere. And we might need all of them. But the good news is that when it comes to soil sequestration, right at the bottom here, it's we basically know today how to implement those practices, and the cost is relatively small. also because it comes with a lot of other benefits, including economic benefits to the producers. And so pointing and trying to do more policy to increase carbon sequestration, it can be a win-win-win. But we are here all biogeochemists, and we need to understand that when we talk of carbon sequestration, we cannot forget about the other incredible ecosystem services the soil provides. And first and foremost, the supporting of the soil biodiversity and the provision of fertility, and in particular nitrogen. So if we want to sequester carbon in soil, we at the same time need the soil to be healthy, meaning that the microbes need to be diverse and transform the organic matter in there and live off of it. And more than anything, we need soil to recycle so that they can provide the natural fertility that we need if we want to stop the use of mineral fertilizer. For sure, there is no benefit in sequestering carbon in soil if we have to supplement that sequestration with nitrogen fertilization, because then it will cost more to fossil fuel to produce that mineral nitrogen than the carbon that we sequester. And so we need to have an holistic picture and understand how do we, bake the cake and eat it too, but leave something there. It's not easy. We need to, of course, and I'll be talking more and more in terms of the inputs. Sorry, I wanted to point to, for sure, inputs are critical to increase that pie, but at the same time, maintaining that diversity and an efficient transformation, but also the output in a way that nitrogen can be recycled, but doing in a way that the input always exceeds the output. That's how we need to think systemically. And so that brings to basically the question that has been at the center of my research program, which is, how do soil organic matter form, turn over, and persist? And so I'll be starting by giving you some example of the methods that we use and how we approach that question, and then we'll go from there. So one thing that I found it very exciting, but that it also has been critical in informing a lot of my research, is the fact that I haven't studied just one system. I worked from the Arctic to the tropics in grasslands, forests, and agricultural systems, and by applying a number of different approaches. For sure, we do more and more a lot of soil, large-scale soil measurements and collect deep soils from a bunch of different ecosystems. We do a lot of controlled experiment, in particularly with the use of isotopes. And of course, we do a ton of lab work where we analyze all those samples. And that's where I want to start. For many people, soil is dirt, and it seems obvious how you process soil sample. But actually, in a world where a lot of people are now measuring carbon in soil, seems trivial for us scientists, but actually it's not. And there are many, many labs that today are measuring carbon, and each of these numbers is one sample. So you should have one number. Look at the spread. Sometimes we send the same samples to a number of commercial labs. We got differences as large as 2 to 3% carbon in the same sample. And when we send a dry and a wet sample, it was humongous difference, because a lot of labs don't dry their samples before they analyze carbon. They do not sieve them and remove rocks and roots. So So we are now going back to the board and kind of doing a lot of training and a lot of speech on how you are supposed to measure carbon. And even more, I don't know how many of you, like us in Colorado, have the problem of inorganic carbon in our soils. There are a lot of labs who have no clue about the distinction between the inorganic and the organic carbon that together make the total carbon, and some of them do not measure inorganic carbon. So if you use labs, make sure you ask what method they are using and challenge them on that, because that's particularly when we go to carbon market, because those are the labs that serve carbon markets. And now we are working on this type of numbers, and we are looking for a less than 1% change in soil, and they can measure over 4% differences within the same soil. So that's a challenge. But for us that work in soil biogeochemists, now, soils are the most complex pool of carbon, and we can spend, a long time discussing all that complexity. And we definitely cannot get there, and particularly when we want to inform management and policy and do models and so forth. But there is some complexity where we need to get. And in my opinion, a very first minimal step is to recognize that soil organic matter is not the same. And some of the soil organic matter is actually of particulate origin, of particular structure, meaning that it's coarser and lighter and come from the structures of plant and the soil biota. Whereas another part is chemically binded to minerals and therefore is heavier and finer, and come either from soluble compounds that directly bind to this mineral or after having been transformed by microbes. And so when we study soil organic matter, besides quantifying the actual amount of carbon in a soil, we like to separate this into pools. But also when we try to understand methods, we want to follow and trace carbon and nitrogen from the input into all these different fractions. And that's why in my lab we do a lot of labeling. I built, as soon as I arrived to CSU in 2008, this labeling chamber, and since then we have done a ton of different experiments there, including now the most challenging is being Arabidopsic. We are working with the Salk Institute now to look at different Arabidopsic genetics, and but we have grown from trees to tussocks to any kind of plant in there. And then we typically either do experiment within the chamber or we harvest the plant material and do experiment in the field without litter bags so that the plants can decompose naturally. We do controlled lab incubation. We take labeled exudates and do exudate incubations. And we also have a passion for pyrogenic carbon and fire and biochar, so we also produce that and do experiment with those. And of course, whether we use this labeling experiment or we go out in the field and collect soil samples, we then do a number of analyses from just stock measurements to pool measurements to trace the carbon and nitrogen into different physical pools, but also do an MR, FTIR. We've tried to do a little bit of nanosims and so forth. So once you have the isotope, you can follow. We try now to do some amino sugar work and, of course, PLFAs and so forth. My label is not high enough to do DNA labeling, but we have done some hydrogen labeling, for example, in PLFA to see the activity of microbes. And so one of the techniques that we use the most is actually a pretty straightforward one in one sense, that is simply the physical separation of soil on the basis of size or density. But the problem that we have when we study soil carbon pools is that it's not that easy to go from a conceptual definition, as you have seen in the graph before, where I say the POM is the structure and blah, 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 I come from this, the MEOM is the soluble and mineral associated, but then how do you separate them? First of all, they can all be either free or inside aggregates. And actually, their presence in aggregates modify the way in which they are formed, the way in which they persist. So you might want to study them either inside aggregates or free, in which case you do not disperse the soil before analyzing them. If you want to have them outside of the aggregates, the first thing you need to do is to disperse the aggregates. And then either do a size separation where you say, okay, all the mineral associated is less than 50 or 60 or 20, depending on the labs. That's another thing we don't agree on, is the size of our sieves. But then you separate by size, the sand size fraction that today we call CHOWM, it was an election in my lab, which is organic matter associated to the coarse-heavy fraction of soil. So this orange thing goes with the POM if you separate by size. If you decide that you separate by density on the principle that the POM is lighter and the MEOM is coarser, this sand size fraction ends up with the finer mineral fraction. And so depending on your calls, and when you decide how to fractionate, you get different results. And so you can decide to not break the aggregates and separate the aggregates. You can decide to break the aggregates and just do by size. You can decide to separate the POM by density and then either have one dense fraction or separate the MEOM from the sand size fraction. Depending on what you get, you might have a different answer. And so why am I getting there? Because many of us are now doing meta-analysis or working across many different labs. And so we put in the same database fractions that had either been measured like this or measured like that. And so it's important that we understand what are we talking about. And in a way, we studied the effect of fractionation on the nature and the chemistry of those fractions. These are results from an FTIR done on individual fractions measured by either size, density, or a combination of the two. And basically, the take-home message here is that the POM fraction is very dependent on the fractionation. And so this is POM fraction and this is POM fraction. If you separate by density, they are really separate aside. If you separate by size, because they are contaminated with the sand size fraction, they fall similar to the MEOM. So the take home here is that if you are interested in POM, you should really do density. If you are interested in MEOM, in the MEOM, you are okay with size. And size, of course, is much easier and less time consuming and less costly. But of course, all these separations are very tedious and time-consuming and costly, and we want to measure thousands and thousands of soils. And so now I'm also trying to work on increasing the throughput of the measurements. I developed a patent for an instrument, but at the time it's still very clumsy and not easy to use. So maybe if I find some company that are interested in developing it further, it might be more appealing. For sure, we are doing more and more work with the FTIR to be able to scale and model the fractions. And then, of course, now we're doing a ton of work with artificial intelligence to try and scale it up at the continent level. And that's the approaches we use as we start fractionating samples across continents. We worked with the JRC and fractionated the Lucas soil, and the NRC fractionated all their soils. The NIA opinion, and of course, we work a lot on meta-analysis to put all this data together. So that's to give you an overview of the approaches that we use. And now I go to the juice part and ask the question. So, applying those methods, what have we learned? How do POM and MEOM form and stabilize? And putting together a lot of the work that has been done in the last 20 years and more, I will show you when we talk about the controls of those processes. Basically, we now understand pretty well that it's very important to start from how the carbon makes into the soil. If the carbon entered the soil as structures, so plant litter, root litter, but also fungal, the structures of soil biota, those structures either persist for too long as free particulate organic matter after fragmentation. They can be protected in aggregates, or they can be depolymerized to smaller molecule and go down. The depolymerization of the structure, and more than anything, the exudates and the leachates from the litter, so everything that is soluble and has low molecular weight, that can either directly sorb to minerals and stay as free or occluded in aggregates and make the MEOM, or can be further depolymerized and mineralized all the way to CO2. And of course, all this transformation go up and down because the microbes in the soils are the one that then recreate polymers or create the enzyme that depolymerize it. So you can imagine a lot of transformation up and down here, controlled in particularly by microbial catabolism and anabolism, or stabilization at any point in time, and of course, the mineralization. And so one of the pathway that I'm most affectionate to is this idea of the two-pathway model. I've done now a ton of experiments in the lab, in the field, one also with some of you here in the audience, and it really confirmed that the soluble input to soil very readily for mineral-associated organic matter, either directly or through first being assimilated by microbes. So what I'm talking about, the first evidence for this mechanism came from a study I did with my first student here in CSU, Jenny Sung, and we incubated in the Konza Prairie big blue stem that was enriched with isotope. First of all, when you put the litter without bags, in 3 years, we reached complete mass loss. And that's because through the end, the litter fragments and doesn't stay in the litter bags, but enter fragmented into the soil and form a lot of carbon. The fragmentation at the end is a very efficient process that forms particulate organic matter. At the beginning, you have the leaching of the soluble and also the depolymerization of not lignin encrusted celluloses and hemicelluloses. And those either directly sorb on mineral, or we found them first in microbial biomass and then on minerals. And so this two pathway has really been something that we have seen over and over. And most recently, this is a paper I just submitted where we looked at breeding corn for deeper roots. And so here in this study, we had 2 different corn varieties. One was more recalcitrant than the other, and we incubated either the root structure or the exudates. And as you see here on top, when we incubate the exudate from these 2 different varieties, the majority formed MEOM, but some also formed POM. That is that evidence for the upper that microbes, you give them soluble, they form their own cell walls that then make POM. So it's not always, our model always go from POM to MEOM. Actually, we do have POM formation from the soluble and the microbes. And then when we added the root tissues, we mostly saw palm being formed, but actually at depth, around 60 to 9 centimeters, that was more efficient, and the most recalcitrant root tissues created the majority of the palm. So again, very clear evidence for that two pathway, and also for the efficient formation of palm at depth, where the microbes are less dense and decomposition is lower. How am I doing with time? What was very exciting is that actually, NSF project are only 3 years. But what I did, I left one more color in the field past the 3 years. And for serendipity, my student was actually in Fort Collins 10 years after. And we went back and collected that remaining color 10 years. So the first paper ended year. And now we have the 10-year project. And guess what? The meum basically stayed the same, whereas the palm largely decreased. But you don't see an increase in meum because the palm decreased. So we keep having, after 10 years, a pretty strong evidence that once they are formed, they are pretty much independent from one another. And so again, it's important, the inputs, but there isn't that humongous transformation and translocation of carbon between the two. And these coefficients of variation also illustrate that the palm and the sensile fraction, its cheom, are much more variable than the meum. But when can the palm persist? In our studies, the palm can persist when it's occluded in aggregates, this gray guy down here. This is a study we did also with the label litter in Canada across a number of different sites. And this is the comparison of the 6 months and the 5-year sample. As you see in yellow, the majority of the meum stays. In gray, the majority of the occluded palm stays over 5 years. In blue, the free palm was basically lost in 5 years. So that also gives you an idea of different formation and how where they are. That tells a lot about their future. But while I showed you that the palm goes and the meum persists, if we look across much longer time frame as the 14 carbon dating can enable us, now you start seeing that yes, the particulate organic matter on average is younger than the mineral-associated organic matter, but their range overlaps a lot. And it's actually span very large range. So you can have some palm that is very old, and you can have some meum that turn over fast. So I don't like at all the idea that, for example, carbon markets are having now to give different prices to palm and meum. That in my mind is incredibly dangerous. Also because the majority of the carbon on Earth, which is in the permafrost or in the wetlands, that's largely palm. And that says we value it less. And actually my personal opinion is that we should value palm more. And as I show you in a minute, the large difference between agricultural soils and natural soils is because we have lost that palm. So depending on how we manage ecosystem and depending on the climate, palm can persist. And also the other thing that we learned from this graph is, yes, separating palm and meum makes sense, but there is a lot more that we need to uncover because from many different ways they do overlap. And so that brings us to the next question. We kind of have understood how those different fractions form, but what controls them? Why do we see that overlap in their permanence if they are so distinct? And that's really an area that has been fascinating working in the last 20 years. Katie Rossi, a former student of mine, just led this synthesis where we had a lot of fun all together trying to synthesize the progress that we made in the last 20 years with all these ideas that were proposed at a certain point and kind of got reinforced and repeated. And more recently, the microbial control is becoming more and more important. And so again, when we come to what controls this soil organic matter formation in pom and meum and the persistence, again, we recognize the importance of separating the input, insoluble and structural, and then trying to follow how those inputs persist or leave, with the microbes really being at the center of everything that happened in the soil. And the thing that here I also wanted to point out is that while we now have a pretty good understanding of the controls, not all of our hypotheses have been either tested or proved right, and so there is a lot more work that needs to happen, and where we're really lacking, and I'll finish my talk about our effort to do that, is to transfer this understanding into models. Our models are still far from capturing the large understanding that we have achieved in the last 20 years. And so this is the framework that I'm working a lot on currently. And it's kind of complicated, so bear with me for a little while. First of all, I'm a biogeochemist. So as biogeochemists understand that when you have a pool, you can change that pool depending on inputs and outputs. And if you want to increase that pool, the output has to be less than the inputs. And so if you put it that way, the question is, which systems are limited by inputs, which systems are limited by outputs, and what are the control on those limitations? And if they are input limited or output limited, what's the structure? What is the implication, for example, for the provision of fertility and for the structure of the soil organic matter? And so if we start reasoning along those lines, you can see that there are systems, and here you can imagine a peatland where the large majority of the microbes are inhibited by the lack of oxygen or by the freezing temperature. There are, of course, you guys teach me that there is always a microbe that thrives in any environment, but I would say the large majority don't. And that results in plant input not being highly decomposed, and therefore the majority of the carbon would accumulate in that pom. On the other hand, you can expect the system, when the pom accumulate, you then have nitrogen immobilization, and so that limitation on fertility. Whereas on the other hand, you would have that you have a limitation on the input, and your microbes basically have transformed the majority of what they get. And so the majority would stay in the meum. And in that case, you might have an open nitrogen cycle because the plant will not be able to uptake all the nitrogen that is recycled. And these things in between. And of course, the subsoil that we are very interested in, you could think of them as being perennially input limited. And then the question also comes on the implication of the mineralogy and texture of soil to retain that carbon. And so, we know pretty well what are the controls, climate, and on the plant productivity and the input. And so it becomes interesting to discuss what are the controls on that output, and what are the output, of course, come from the controls on microbial activity. And so again, what I was alluding before is that when microbes are limited by or are inhibited by the lack of oxygen or or the cold temperature, you can expect that you have high persistence of carbon, but this time the majority will be in palm, and actually those soils will have a lot of carbon because microbes have not decomposed the inputs over time. But the bad news is that carbon will be very vulnerable because if we drain a wetland or if the permafrost form, now microbes will be released from their limitation and can go at it. On the other side, if the microbes are actually limited by access, so the carbon, they cannot get there, maybe because it's occluded in a very fine aggregates or because it's bonded to minerals, at that point, the microbes will already have decomposed everything that is accessible. The palm is largely gone. The majority is in the meum. There is a lot of very high nitrogen because it's been mostly microbially transformed. But the good news is that that carbon, we could expect it to be less vulnerable because now it's harder to mobilize carbon that is minerally associated. But of course, if we start doing hard tillage, plowing, and also the fallow, if we do not provide more inputs, microbes will find a way to get at it. So it's not like it's never going to be decomposed, but it's less accessible. And so when we think about health, we really would like to have our soil in that stage with that mixture of palm and meal. And anyway, we have now starting to do a lot of work compiling data from across the world or the continent and applying structural equation models to go at those hypotheses. And we are finding quite a good confirmation of them. This is a study that a former PhD of mine, Paige Hansen, published earlier this year, where she looked at the structural equation model on a large data sets of soils from all over the world. And she found what we had suggested in the hypothesis, that palm is really controlled by control on decomposition. So temperature, pH, whereas meum is mostly controlled by control on inputs and control on stabilization. And here we only had texture, but in a minute I'll show you that there is more than just texture. And this hypothesis was also confirmed by when we started looking at how do the soils across continents change and across different ecosystems. These are analyses we did in the Lucas database in Europe. And so what we notice is that agriculture always have less carbon per unit land, and the majority of it is in the meum fraction, and they have relatively low carbon to nitrogen ratio. Whereas if you go to more natural systems like forests or grasslands, you have a more balanced distribution between palm and meum and a higher carbon to nitrogen ratio. And so when we did that globally with Paige, there is this index that we call the F-meum. So the proportion of carbon that is in the meum fraction versus the total. And this is actually an index of not health. The highest the F-meum, the more the soil has been disrupted and lost their palm. And that's what you see in agriculture. And that's why I say for the young in the room, it's good to analyze different types of soils. The problem with a lot of people that have only looked at agricultural soils is that they don't even know what a soil looks like. Because an agricultural soil has been highly transformed by our management and is a soil where more than 80% of the carbon is in the meum fraction. Whereas natural soil have by far more of a share in palm, in particularly those wetlands and peatlands where they have by far more. And so you can think almost as a scale of decomposition inhibition, whether you have more or less of that palm. And I said, like, for the meum to accumulate, you need the inputs and you need the capacity of the soil to store it in minerals. And we are now appreciating more and more, and I know that several of you work on that, is that it's not just texture, of course, but it's not even just pH. as this graph show, a lot is in the presence of iron, aluminum, calcium, and magnesium. And we propose this matrix capacity index that has all of it and is a better predictor of total carbon and milk in particular than just texture. Now things are getting really complicated even for how I like, but Jocelyn Lavalle is really good at doing structural equation modeling and putting a lot of parameters there. And the other thing, that this is a continental US scale of thousands of soils that we have analyzed from the NICS database, or some with real fractionation, and some we have predicted with the FTIR. And if we put all that data together, what has been becoming very interesting is that hierarchy of controls. And so aridity is really an overarching control, to the point that the structure of controls of POM and MEOM depend on the aridity index. And so when things are dry, basically productivity becomes key. You need more input. That goes back to that in and out framework. So when it's very dry, you do need those inputs to control POM and MEOM. But that's not the case. There is no MPP control on POM and MEOM in wetlands. There instead is mostly driven by the capacity of the soil to store it and disturbance. And so we really, when we want to start understand these things, we really need to think about those larger control. And so I hope that this information is really enable us to say something to management and modeling. We are seeing, when we do meta-analysis on regenerative ag, that actually with change in management, we can increase both POM and MEOM, particularly in the topsoil. And it's important to see that, yes, POM needs to be a target because it's where the health of the soil rely. And it changed more proportionally, but since it's a little pool, actually affect less the change of the total carbon. And so we also need to change the MEOM. What I'm very excited about as an ecologist is that bringing animals in agricultural land, this livestock integration, is really one of the most effective things, but we have very little numbers, so we need to study that more. And that brings to grazing land. There is a lot of potential in grazing land with modifying management to increase carbon sequestration. And I particularly work on the grazing management and on rotational management. And we are now following this concept that we worked on with Paige Stanley and others in my labs, where we are really looking again at the ecosystem scale, how grazing management affect the ecophysiology of the community, and that in terms affect the carbon pools. And we are getting some nice results, like with modifying management, we are seeing increases in carbon, particularly with adaptive management. And one of the largest projects currently in my lab is this that is co-funded by FFR and Noble. And we are working all across the US, both on farms and in experiments to get at that overall things. And so by doing conceptual work, by doing on-farm study, we are now trying to put it all together in the development of a process-based model. That's work that I'm doing with Yao Zhang. I'm not a modeler, but I'm really getting excited to work with him and putting all what I told into this process-based study. We have developed it on the neon grasslands, doing a pretty good job of representing pools and flaxes. We have developed for croplands, also working on Mary flax sites, and we are having trouble with tillage. We are now trying to do aggregates and tillage. We are putting a lot of effort in representing grazing management and then trying to use this tool to predict the future and say what levels of grazing can be most effective. We are seeing that modifying grazing can have a significant outcome and increase carbon, but the total carbon that we are seeing are actually less than what the industry would like to see for those practice. I like to close by saying that at CSU, we funded the Soil Carbon Solutions Center. I'm now less involved in it, but we are trying to start a consortium that is going to be free for scientists and NGOs and has a membership fee for industry to have access to the data and the model. And with that, of course, I'm only good at talking and bringing money in, and all the work I presented is done by the amazing team that I have the pleasure to work with and mentor, and of course, to a number of collaborators. And with that, I thank you and for the time.